Our first student who is going to be presenting is Kathryne Chamberlain, or Kate. She will be a senior at Canyon Crest High School coming up this year. She did not want to specify what schools she's interested in. There were apparently way too many to list, so she is going to be working diligently on applications for many schools across the country. And I know the rest of you will be doing this as well, college applications for those of you who are getting ready to do your senior year. That's going to be something you're going to be working a lot on, and we're happy to help you with any of that if we can. But she's definitely interested in a science major, and part of her experience this summer was figuring out what kinds of things in science she liked. As Ellen said, one of the things that all the students experienced was a very, very steep learning curve early on. The first week when we met with them, at the end of the first week, their eyes were about this big. And they all just looked at us like, wow, what did I let myself in for? But as Ellen said, they rose to the occasion wonderfully, and you were about to see just how great they all managed to do. I asked them to tell me what were some of the things that they learned during the summer, and Kate was a particularly good example of what most of them had to say. Even a failed experiment is a step toward success, which I like that. Kate.
Hi, I'm Kate Chamberlain. I spent this summer interning with John Naughton in Dr. John Young's lab of the NOMA Center for Immunology and Microbial Pathogenesis. I spent this summer researching HIV-1's interaction with TAM receptors. To give some background on the Young laboratory, it's divided into two parts. Part of the lab is studying anthrax and the other part viruses. Specifically in the anthrax side of things, scientists are looking at toxin entry and pathways and different receptor decoys and inhibitors for these processes. On the virus side, the focus is mainly on the human immunodeficiency virus, or HIV, and specifically HIV cofactors and restriction factors through gene inhibition with siRNAs. Another focus of the lab is TAM receptors, and they've shed light on the importance of TAM receptors in HIV infection, which is the focus of my summer research. For some background on HIV to start, HIV follows a replication process that has been defined very specifically and pretty definitely over the past few years. HIV begins by binding to a cell through different receptors and co-receptors. It will then fuse with the membrane and release RNA into the cell. Because it's a retrovirus, HIV carries RNA with it instead of DNA, which requires reverse transcription to turn the RNA into DNA to then be integrated into the host DNA through integrase. After the viral DNA has been integrated into the host DNA, it then undergoes transcription to be turned back into RNA, which is eventually assembled with different viral proteins and buds from the cell to become the virus. After maturation, the virus is able to infect and replicate between other cells. So because of this process, current research mainly focuses on CD4 receptors and CCR5 and CXCR4 co-receptors, which is the entry of HIV into cells, as shown by this picture. However, recently, Dr. John Young and Dr. Greg Lemke of the Salk Institute have actually shown that HIV hijacks TAM receptors in mice cells to increase their infectivity efficiency. This presents TAM receptor inhibitors as potential therapeutic medicines for slowing the spread of HIV. A little background on TAM receptors. TAM receptors, as you can see in this picture, help to bind apoptotic cells, right here, which is a cell that has a signaled, or like a controlled cell death, through ligands and receptors to the phagocyte, which is a cell that's in charge of consuming and clearing the apoptotic cell. The TAM receptor family includes tyro3, axyl, and mer, which commonly bind to the ligands Gas6 and protein S of the GLA domain. In a normal functioning cell, there's an enzyme called a flippase enzyme. This is in charge of keeping the phosphatidyl serum on the inside layer of the cell membrane. However, once the apoptotic cell has reached the time to die, that enzyme will stop working, allowing the phosphatidyl serine to move to the outer layer of the cell membrane. When this is exposed to the extracellular space, the ligands Gas6 and protein S are able to connect to the phosphatidyl serine, which enables binding to the TAM receptors. Because this only occurs when the cell is ready to die, this allows the TAM receptors to activate the cell to endocytose the cell, or take it in to clear it from the bloodstream or wherever the cells are. Because the apoptotic cells are cells from our own body, or autoantigens, you do not want to create a full-blown immune response against the cell. So to limit this possibility, the TAM receptors result in an upregulation of SOX-S1, or a suppressor of cytokine signaling protein 1. This, in turn, leads to an inhibition of interferons, which would normally fight an infection. Because of this process, TAM receptors are extremely important in maintaining homeostasis throughout your body and preventing your body from autoimmune diseases. As shown recently, this relates to HIV because when HIV buds from the cell, it encloses itself in the cell membrane with the phosphatidyl serum and all. Because the virus is much smaller than the cell, it only takes with it the proteins and the RNA needed to infect and replicate in other cells, which does not include the flippase enzyme. Because of this, the phosphatidyl serum can then be exposed on the outside of the cell just as occurs in an apoptotic cell. When this happens, the virus can then bind to the Gas6 and protein S ligands and then connect to the TAM receptors to activate them inside a phagocyte. When the TAM receptors are activated, they read the cell just as they would an apoptotic cell, which in turn will lead to endocytosis, so the TAM receptors will take the virus in while shutting off the immune response. Because of the interferons that are blocked by the TAM receptors, because they're often antiviral agents, we would expect that this would lead to an increased viral infection. We've hypothesized that virus grown in media that contains all of these GLA domains will have a higher infection capability than the virus that's grown in media depleted of the GLA domain, because complete media enables TAM receptor activation, which will lead to a decreased immune response. We will examine this process in HeLa cells and THP1 cells. We chose to use HeLa cells and THP1 cells as our model cell system because they're both human cell lines. All previous research, as I said, was done in mice cells, and so the next logical step to moving this process into human cells would be to look at human cells. So both HeLa cells and THP1 cells are immortalized cells, meaning they'll continue to grow and divide if they're in the correct media. However, more specifically, HeLa cells are adherent cell lines, which means that they're much easier to work with because they'll stick down to the plate when you're using them. And so these make a great starting tool for someone who hasn't had a lot of background in tissue culture. While THP1 cells are a little harder to work with as they're in suspension, they're a closer representation of macrophages when they differentiate. This is important because macrophages are a major site of HIV infection in our bodies. These cells are also close representations of primary human cell lines, but being a 17-year-old high school intern, I can't use primary human cell lines. Initially, our plan was to grow the virus. It's known as the VSVG pseudotyped HIV1 NL43E-R+FF Luke virus. What this means is the envelope gene is deleted from the virus, and we add in VSVG. This changes the envelope, which is known as pseudotyping. In previous experiments, this was important because mice cells don't contain CD4 or CCR5 receptors or co-receptors. So wild-type HIV can't infect the cells. But when we pseudotype them, it allows the cell to infect mice cells, it allows the virus to infect mice cells as it would in human cells. For my sake, although I'm working with human cell lines, pseudotyping the virus makes it much safer for experimental use. We also replaced the NEF gene with luciferase, which allows us to read out the amount of virus in a sample after we infect it. We would grow the virus in both complete media and GLA-depleted media, equalize the concentrations, and eventually infect the HeLa cells and repeat it on THP1s. To do this, we grew the cells in tissue culture, which just consists of growing the cells in a plate with their respective medias. And the first experiment we completed was a transformation. This experiment refers to the introducing of different nucleic acids into a bacteria cell and allowing it to amplify and then purifying the sample to create a stalk of a plasmid, or the specific DNA you want to use to create the virus. After we created the plasmid, we then transfected it, which is shown in this diagram, into 293 T cells. So specifically for me, the NL43 plasmid that we created through a transformation, we then used in a transfection to produce the virus that we needed. After growing the virus, we then infected the HeLa cells and read out the results. As you can see here, these graphs show the relation between the luciferase amount, which is proportional to the amount of virus, with the virus dilution. The Salk virus in both studies which is a virus that's created here at Salk, has extremely high concentrations of virus. However, the virus in study A that I transfected in complete media did show signs of virus, but significantly lower than the Salk virus. And the virus that was transfected in media depleted of the GLA domain shows no signs of viral presence. In study B, we did the same thing with different plasmids, and the two viruses that I transfected in complete media and depleted media did show indication of virus, but in extremely low concentrations. Because these results weren't as we had expected, we decided to run a P24 detection assay, which is another method to indicate the amount of virus present in a sample. Known as an ELISA, this is an enzyme-linked immunosorbent assay, which is a way to detect different proteins. We specifically tried to detect the P24 protein, which is produced by viruses. And to do this, you add the sample to a plate that has the capture antibodies already at the bottom of it, which will then bind to the protein. And using two other antibodies that will bind to each other and the protein, you can then use a substrate to produce a color change and again read out the viral concentration. So from doing this, the results actually indicated similar results as we had seen before. In the first study, the virus that was produced in complete media shows signs of virus, but still significantly lower than what the Salk virus would produce. And in study A, we didn't use the Salk virus because we were confident that there was plenty of high viral concentration. We also used the GLA-depleted virus, which again showed no signs of virus. In study B, the Salk virus had high concentrations, and both the two amounts did indicate virus, but in very low concentrations. So although this validated our results, we still didn't know why the GLA-depleted transfection was unsuccessful. So we then decided to do a restriction digest. This is the process of using enzymes to cut a plasmid in specific locations in order to use predicted sizes and figure out if the plasmid is what we think it is or if it has cut out certain parts and recreated into a new plasmid. So it wouldn't work. For example, we used Sal1, which cuts the enzyme in one place, and we would expect to see a result that indicates a plasmid at size 14,824 base pairs. We also used an enzyme known as APA1, which cuts the plasmid in two places, so we would want the results to indicate a piece of plasmid that's this size, and one as well that's a little bit bigger with this size. In order to read the results, we run the plasmid onto a gel. By loading the plasmid into the gel into different wells, you can use electricity to bring the plasmids, an electric charge to run the plasmid down the gel. The longer plasmids will run slower and the shorter ones will run faster. AVR2 is one of the enzymes that we used, and the expected band positioning is shown by these asterisks. As you can see, plasmid 1 had band sizes that matched those perfectly. This was a clear indication that plasmid 1 is the correct NL43 we were expecting to use. However, clearly plasmid 2 is not the same virus, although we were expecting, not the same plasmid, although we were expecting that they were both NL43. Because of the different band patterns as seen in plasmid 2, we were able to conclude that the GLA-depleted transfection was probably unsuccessful because of plasmid recombination. Even though we now understood why the GLA-depleted transfection did not work, we still had inconclusive results regarding the TAM receptor interaction with viruses. So we decided to take a parallel approach. This included using a pharmacological inhibitor of TAM receptors, known as SBNG10, with the viral infections. This is a chemical that will inhibit the function of TAM receptors, which doesn't allow the virus to bind to these receptors. We would expect to see lower infectivity rates because the TAM receptors are no longer able to shut off the immune response within a cell. So the cell can provide some resistance to the virus infecting it. This also leads to a potential medicine in slowing the infection rates of HIV, if successful. As you can see with our results, clearly the starting position for the virus, for how the virus would infect, is decreased with the increasing concentrations of the compound. This linear decrease is a clear indication that the compound does affect viral infectivity. Also, the viability of the cells shown on the right indicates that the cells do remain stable. And although there's a small decrease at the end, it's only barely statistically significant, and it indicates that the cells still remain viable despite the little decrease. We did repeat this experiment again, And the results, although less nice looking, they do indicate a decrease in the amount of infection from the virus with the higher increases of the compound. And although the viability does remain below the line that we would have expected, that we would have wanted the cells to remain at, they're still within the 65% threshold to indicate that the cells are still viable and non-toxic. We repeated this experiment on THP1 cells, which was not as successful. The compound seemed to have no effect on the infectivity of the virus in the THP1 cells, and the viability did remain stable. However, we think that because the amount of luciferase is much lower, that increasing the infection time with the THP1s would make this more successful of an experiment, because as I said earlier, the THP1s are harder to work with, so plating them out evenly and infecting them with the right amount of virus and giving enough time can be more complicated. So when we repeat this experiment, we would give a longer infection time, maybe 48 or 72 hours instead of 24 as used here. So overall, throughout this summer, we began by looking at the TAM receptor interaction with HIV. And our first attempt was to look at the virus and manipulate the virus to either have the GLA domain or not have it in order to infect the cells. Because this wasn't as successful as we hoped, we again made a second attempt, and this was to use the compound to manipulate the cells before we infected it with the virus. And because this was more successful, it encourages us and gives us more promising results for the importance of TAM receptors in viral infection. In the future, besides optimizing the viral transfections in this experiment, we're looking to see if this is a common mechanism with many enveloped viruses. This process, unlike looking at CD4 receptors and CCR5 co-receptors, which are extremely HIV specific, this is more non-specific. We would expect to see that this interaction would change with Ebola, hepatitis C, influenza, and West Nile virus, which are all being studied by our collaborators across the country. Also, it's important to compare wild-type HIV-1 studies with the VSVG pseudotyped HIV-1 that I used. This is because the entrance of the wild-type and the pseudotype virus are slightly different. As you can see in this diagram, HIV that's not pseudotyped will enter the cell through membrane fusion, while HIV that is pseudotyped will enter the cell by pH fusion. Although the steps that remain after this process are completely the same throughout the cell, this small change may have bigger results or bigger impact than we would think. Therefore, it's important to show that the VSVG pseudotyped HIV is a reliable representation of the wild-type HIV. I would like to thank my mentor, John Naughton, principal investigator, Dr. John Young, the educational outreach coordinators, Donna Mapston and Ellen Potter, the LSSI coordinator, Erica, our teachers at the boot camp, Dr. Sanda Slivka and Sharon Price, LSSI scholars, the Salk Institute, and the Young Lab. Yes, all of you. Thank you.